Okay, we're gonna do chapter 5 right now. Flip to chapter 5. If you haven't finished, you can screenshot this thing. Strand means a long substances, a line. Strand. Okay, chapter 5 right now. Diagram 2.1 shows the stages in the reaction of enzymes amylase. It says this is an amylase enzyme. Okay, the enzyme right now is amylase. Between P and S, which one matches with the diagram? P matches, right? Because the shape matches. Every enzyme, there is this part here. What is this part called? This is what we call as, correct. It's called active site. Every enzyme, they will have their specific site. We call this as active site, active binding site, correct? They have to match. Only highly specific they can match together. So what is the question asked again? They ask, based on diagram, name complex K. What is complex K over here? This complex K, when an enzyme and substrate join together, it's called enzyme substrate complex. Very good. It's called enzyme substrate complex. Okay, you can also write they are amylase starch or amylase substrate P complex. Also correct. If the enzyme used is a sucrase, what are the product produced? Okay, right now they give you another assumption. They say that right now this is a sucrase enzyme. Sucrase enzyme, they will break down, cannot write substrate enzyme complex. Enzyme substrate complex, no substrate enzyme complex, oh my dear. Correct. When the enzyme used is a sucrase, the answer is glucose and fructose. Very good. This is glucose and fructose. Glucose and fructose. Remember GFX. GFS, you turn the X into S. GFX, this is how you remember it. If it's glucose and glucose, it will be maltose. Glucose, galactose is lactose. And next, based on diagram 2.1, state one characteristic of enzyme. Okay, the keyword is based on diagram. When we say based on diagram, means things that we can see. We call this as based on diagram. What we can receive in this diagram? Can you see their catalyst? Can you see their speed up reaction? Cannot see, right? So if it cannot see, we can only write what? We can see that they are highly specific, correct? The enzymes reaction are highly specific. And enzymes are not destroyed at the end of enzymatic reaction. So we're going to say enzymes reactions are highly specific. Next, you can say enzymes are not destroyed at the end of enzymatic reaction. Enzymes are not destroyed at the end of enzymatic reaction. So if you want to write based on diagram, I will suggest you to only write these two things, guys. Because these are the things that we can see from a diagram. You cannot write enzymes reaction are reversible because usually diagram, they don't show you double arrow. But this is the two safest answer to write. State the type of enzyme for amylase. Okay, what is the type of enzyme? We only have two type of enzyme. We call this as intracellular and extracellular enzyme. We only learn two type of intracellular enzyme. Anybody remember what is the example of intracellular enzyme? What is the intracellular enzyme? If diagram they circle one binds with the enzyme, do we say that they are affected by the enzyme inhibitor? Circle one bind. If they binds with the abnormal shape, it means they have to be inhibitor. Okay? Correct. It's called lysozyme and hexokinase enzyme. Very good. Lysozyme or hexokinase enzyme. There's only two enzymes, guys. Produced in the cell, used within the cell. There's only two enzymes. The rest of the enzyme that we have learned, no matter whether they are lactase, lactase, amylase, maltase, everything else, it will be extracellular enzyme. For student, the enzyme substrate also be based on diagram because they told the enzyme is an amylase. The enzyme is an amylase. Yes, the enzyme is an amylase. Then shouldn't the enzyme substrate also based on diagram? What do you mean? Means this thing is highly specific. Only the correct shape can bind with the correct pattern. This is another question. Not quite related to this actually. I want to say it's just another question. They say assumption of if what? You said this is if, but right now it's amylase, right? The diagram here shows it's fixed. So these are the two answers that you can write. I don't know whether you are asking whether to write which answer or not. By the way, back to this again. This is a type of extracellular enzyme. Amylase is a type of extracellular enzyme. The naming complex K. Uh-huh. If you want to write the complex K, you can write enzyme substrate complex. You can write amylase substrate P complex. Also can. So it's based on you because this is a general name. You can write specifically also correct. I mean, it's not wrong to write this. It's not wrong to write amylase substrate P complex. Okay, substrate S is not able to combine with enzyme amylase to form complex K. Explain why. Why S cannot bind with amylase? Because what? The shape doesn't match. When the shape doesn't match, you cannot combine together. Why? Because we mentioned before, right? Enzymes reaction are highly specific. They can only bind with a specific diagram, which is it has to be matched. The specific substrate can match with the specific active site of enzyme. So in sentence, we can say enzymes reaction are highly specific. Substrate X does not have the shape that can bind with active site of amylase. That can bind with amylase. So usually when we explain our answer, we will also use the characteristic of enzyme. 2.2 shows the action of molecule Z on amylase. What is the effect of presence of molecule Z on amylase activity? The diagram doesn't fit at all, but they match together. What is this molecule Z called, guys? Can we see it does not match the shape of active site? Can also? Correct, they are inhibitor. Inhibitor is like a third party in the relationship. You and your partner happy together. Suddenly got one third party coming in already. That's called molecule Z. Can write not compatible for the previous question? Yes, you can. You can write not compatible as long as you have a similar meaning. Can they will compete, correct? If you look at all of the drama that you watch, right, all of the third party, they have to compete with the girlfriend, boyfriend. Okay, when they compete, who's gonna win? The third party gonna win. All the drama is the same one. The third party gonna win anyways. Will they have a happy ending? Never. Never happy ending one. This one also never happy ending. The enzyme reaction will stop because they are not meant for each other. Whatever you compete back is never good for you guys. It's inhibitor. So we have to say molecule Z is an inhibitor. And then you're gonna say substrate P compete with molecule Z to bind with active site of enzyme. Then we're gonna write active site of enzyme are occupied by inhibitor that causes rate of enzymatic reaction to stop or decreases also can, stop also can, up to you. Teacher can say molecule Z have higher affinity towards enzyme compared to substrate P. Actually can lah, but usually when we use the word affinity in biology, we only use in the carbon monoxide. but the meaning is okay. The meaning is the correct meaning here. But can as long as you write the word, because usually for enzyme we use the word compete. In carbon monoxide we use the word affinity. If your teacher are okay with this word, I think it's fine, but some teacher they want you to write the word compete. Can you write less products are formed? Of course you can, but because the question is only 2 marks lah. If you need 4 marks, you can write lesser products are formed. Can ya? F. Why are apples that have been boiled after they are peeled do not change the color to brown? Word here is called pale, boiled. Boil means what? Boil means higher temperature. We call this as boil. Every apple that turns into brown because of an enzyme. When they have enzyme, they will oxidize. Right now, if it's higher in temperature, means what? Next Monday class still from 4, of course. Correct, they will denature, yeah? Denature, okay, denature. So we have to talk about denature. So when it denature, no matter whether it's high temperature, your apple will not turn into any color anymore because it's not optimum temperature. So we have to say apple contains enzyme that transform the color of apple tissue to brown after it is peeled and left for a while. Next, denature in Chinese. Chinese, I don't know the exact word, but in Chinese, in normal English, it's called changes the original shape. 改变它原本的形状. Only enzyme can use the word denature. But if you ask me the specific word for denature in Mandarin, I also don't know. But it means changes the original shape. So the enzyme has to be 3D shape. So if you put it into not optimum condition, it becomes abnormal shape. So they cannot function anymore. Denature means they cannot function anymore. Changes the shape and cannot function. I don't know what is the specific word for Chinese lah. 哇，这个考难我了，这个东西。Okay, so we're gonna say if apple is boiled, enzymes becomes denature, causing apple not to turn brown. So I write the meaning down from denature lah. Denature means changes its 3D structure. 3D because enzyme is 3D in shape. So that's why it's 3D structure. Then it cannot function anymore. It cannot function. It's called denature. Can say enzyme changes the shape. Okay, if you want to write changes the shape, it's usually the explanation of it. We will always choose the word denature. Okay, ready? Everybody okay with this? Okay, you raise your hand, okay? Wait for 10 of you. If you haven't finished, you screenshot the answer. Who has influenza? When does their chemical bond break? A high temperature. Because pH value don't break the bond. pH value only changes the charges at the active site. Wait for 10 of you. 7. What is the difference between extra and intra enzyme? Extracellular enzyme means they produce in the cell, they use outside of the cell. Cell. Okay? Produce inside, use outside. Intra means produce inside, use inside the cell. Like lysozyme, it can only be found in the cell. Your amylase is outside of the cell because you use your saliva is outside of the cell already. So it's called intra and extracellular enzyme. Okay, next question. Bio kertas 1圈圈几嘛？上次你没有考 full paper. Huh? You are form 4 or form 5, my dear? Form 4, I don't have paper 1. Okay, next. 3.1 shows the application of enzyme in dairy industry. So they ask you enzyme K. Oh, denature 叫变形 ，OK， 改变它的形状 changes the shape, correct? So name enzyme K. What is enzyme K? This is in the dairy product. Dairy product is what? Dairy product means this one has to be lactase enzyme. Very good, because you want to produce a lactose-free milk. This is a lactase enzyme. Lactase enzyme, yeah. State the technology used in this industry. What is this technology? This technology, okay, as long as the human changes it, as long as the involvement of human, anything that human changes the enzyme, we have a name. What is the name of it? Man-made technology is called what? It's called immobilized enzyme technology, correct? Yeah. Immobilized enzyme technology. Immobilized enzymes technology. Alginate beads are a type of inert substances. Explain how the alginate beads helps the activity of enzymes K. Now, how do we help the activity of enzyme K? If you don't know what is this alginate beads, it doesn't matter. You only have to remember what it's called inert substances. Can immobilized enzyme be reused? Yes, you can. You can reuse the immobilized enzyme. Why? Because inert substances means that they have a higher resistance. When you put it into a different pH or different temperature, it will not denature. Everything remains the same. So that's why this immobilized enzyme, it can be reused because it will not denature. First, it will not denature. They have a higher resistance towards temperature and pH. So if you think about it, right, normal human, our optimum temperature is 37 degrees Celsius. Outside body is up to 40 degrees Celsius. As long as they are more than 40 degrees Celsius, the enzymes will denature. Right, this is what we learn, right? 40 and above, enzymes denature. But right now, because we changes the enzyme, even when it up to 40 degrees Celsius, the enzymes will not denature. So enzymes can be reused. Imagine we have pH, like we have amylase. Oh, sorry, maybe not amylase. Maybe we say talk about pepsin, your stomach pepsin. It will only works in pH 2. But if I take the pepsin, I put it into neutral condition, the enzymes will also not denature. Why? Because they added with these inert substances. So remember, inert substances, it will have a higher resistance towards your pH and your temperature. Enzymes remains the same, so it can be reused. Okay, it's called immobilized enzyme. As long as human changes the enzyme, it means immobilized enzyme. In Chinese, okay, Chinese means what? Chinese means OK，immobilized enzyme 呢，就是它不会改变形状的。平时呢，我们的温度只可以到可能外面是40度，凡是超过40度呢，就会 denature。现在如果你凡是超过40度的话，它都不会 denature 的，因为这个 technology 呢，就是为了让它不改变它的形状。不管你的温度过高，你的 pH 不一样的话，它也不会 denature 的，它的形状都会是一样，我们一直可以重复用它。就是人改变的 enzyme，这个是它不是天然发生，它是人改变的，或者 immobilize 啊。If you write good, but the Chinese too strong here. I also don't understand what you are trying to say in this Chinese word. Okay, can I? So now you understand what is immobilized enzyme? Human changes it. 只要人改变的都是 immobilized enzyme 啊。Can I? Okay, guys, don't worry. Whatever I say in Mandarin is whatever I say in English. Okay, BM aku tak boleh. English and Mandarin saya boleh. Okay, so we're gonna say to increase resistance of enzyme towards changes in factor such as pH or temperature. The enzyme remains in same position throughout enzymatic reaction. Then we can say enzyme can be reused repeatedly So for this one, you can repeat to use the lactase to hydrolyze the lactose. For example, I can say lactase can be reused repeatedly to hydrolyze lactose. So whenever they ask about immobilized enzyme, you write the same answer, guys. This is the same answer for all immobilized enzyme question. Okay, just repeat this, okay? Only the bracket you cannot write lah, but the rest you can write. No matter whether it's lactase whatever lah, as long as they ask about inert substances, immobilized enzyme, you just repeat the same answer. Can write remains, enzymes remains unchanged throughout the reaction. Can, that will be your second answer lah. Can. Okay, anybody okay with this? Everybody okay with this? Okay, you raise your hand, guys. Okay, now done already. We're going to look at the next question. 5.3 shows the extraction of agar from seaweed. Enzyme immobilized technology is used in various industry applications such as extraction of agar from seaweed. By using an example of immobilized enzyme, explain how agar can be extracted. Again, the question asked about what? They asked about immobilized enzyme. Right now, they give you what? They give you seaweed, agar seaweed. Seaweed is a plant cell. So if it's a plant cell, means that is what enzyme. As long as they are plant cell, the enzyme used has to be the same. It is known as cellulase enzyme. Because plant cell is made of cellulose, the enzyme used has to be cellulase. Can you give me another application of cellulase besides making into agar? Any answer? What is another application for cellulase? Apple juice is with cellulase and pectinase. So apple juice has to add with the word pectinase. Now I'm very sad, my Form 5 student. I teach you like 2 weeks ago, my dear Form 5. What is the function of cellulase? Another example of cellulase in application. Not food one. Is to use in detergent, correct? Detergent is to soften your cotton shirt. So we can use cellulase to soften the cotton shirt. I want to cry already, my Form 5. I just teach you 2 weeks ago. Now your turn. You all don't remember the answer. Okay, so we talk about it. Remember, cellulase can be used in this. Okay, can be used in any plant cell as long as they are plant cell. No lah, got Form 5 in this class also. That's why I asked the Form 5 I just do the same question. This class has Form 4 and Form 5. Got 2 form here, okay? Okay, anyway, how do we explain the answer here? To explain your answer, it's same as just now. How do we write the answer? We have to say they add with what? The cellulase is added with inert substances. What is inert substances? Here, inert substances. Your answer is the same. You write the same answer. But right now you talk about cellulase. You change the last sentence. You talk about cellulase. They will hydrolyze cellulose to extract the agar jelly. Okay, now we have to write in sentence. We have to write the enzyme used is cellulase. Cellulase combined with inert substances. Inert means insoluble, cannot be changed. To increase resistance towards change in pH or temperature. And you're going to write the last sentence. Or if you want to write, it remains the same, also correct, but because it's only 3 marks, then I talk about the cellulase. Cellulase break down more cellulose into glucose. So next time you want to just take a smart guess. smart gas plant, write the word cellulase enzyme. All immobilized enzyme technology uses inert substances. Yes. All, everything. We have to add with inert substances. Okay, in the baby food industry, protease and lipase enzymes are important in the production of baby food. Explain why. Protease and lipase. Can you tell me what is the function of protease here? Protease is to hydrolyze what? Protease is to hydrolyze, correct? Is to hydrolyze protein into the smallest molecule, right? So actually the sequence of breaking down protease is this one. Protein to polypeptide to dipeptide to amino acid. Okay, usually in question which is for digestion in Form 4 Chapter 9, we have to write a specific enzyme because protease is a general name. The example of protease to break down, remember as PTE. What is a PTE? Pepsin, trypsin, erepsin, okay? Pepsin, trypsin, erepsin, PTE, correct? It's called PTE enzyme. So the purpose is to break down into the smallest molecule so that the baby can absorb the food easily. What is the function of lipase? Lipase is to do what? Lipase here, function is to break down your lipid into what? Into fatty acid and glycerol, which is also the smallest molecule. Into fatty acid and glycerol. So it's easier for baby to absorb the nutrients because baby cannot digest. Baby don't have a lot of enzyme. Actually, baby and elderly, both of them, they have reduced in enzyme when they are growing older, well, when they are too young. So they need to break that into the smallest molecule. So you're going to say protease and lipase can hydrolyze protein and lipid into monomers. The smallest molecule are called monomers. The baby can easily consume and, or you can say digest or absorb the food. Okay, can? Done with this? Okay, can okay, now done already. Next one. When produced in industry, apple juice is clearer compared to the homemade apple juice. Explain how pectinase enzyme is used in the production of fruit juice. Okay, pectinase. The moment when it comes to pectinase, okay, it has to be apple juice already, confirm one. So what is the function of pectinase? Because the question already mentioned, this pectinase enzyme is to make what? To make the apple juice clear. That's what the question says. Apple juice is clearer. What is another function of pectinase besides making it clear? Is to more juice, very good. Increase in production of fruit juice, very good. So there's two functions. So number one is to make apple juice clear. Second one is to make sure that they can increase in production. Remember, when you see the word pectinase, it has to be this two answer. The only two answer for this now. So you have to say, and pectinase is applied to break down apple juice. To break down not apple juice. Apply to break down apple, apple pectin. Hence, it make apple juice clear. Then we have what? Pectinase also increases production of apple juice. Also increases the production of apple juice. Pectin, it can also be found in the cell wall. It is to make cell wall also. Apple pectin is to make the cell wall. Actually, plant cell got pectin, also got cellulase. Increased production means that they have, it is what it says, means that they can produce more. So if you use it at home, right, if you make apple juice at home before, right, you'll notice that when you break the apple juice into a juice, right, the leftover fruit flesh, they still have a lot of juice. So when you're using a pectinase enzyme, they are trying to extract all of the juice out from the apple. So you are able to increase the production of apple juice. It will not produce suddenly, but it's in the apple, just that they can extract more fruit juice out lah. Okay, diagram 3.2 shows a type of plant used by housewife to cook meat. Wrap the meat slices in a young leaf and marinate for several hours. Marinate is washed before cooking. Explain why the method is used in cooking of meat. Okay, this is a papaya. Papaya or pineapple, answer is the same answer. So if you want to write the enzyme, write the easiest answer. Don't have to write papain or bromelain. You write the easiest answer, which is protease. Don't make your life difficult. Just write protease, guys. You write papain, they have no extra mark. You write bromelain, your answer is wrong. Just write protease. Papain is correct, guys. Papain is correct, yeah. But if you want to be safe, I will suggest you just to write protease because protease definitely is correct. So first, you're gonna say the enzyme used is protease. The papaya leaves contains of protease. Because the question says leaf, by the way, if you notice young leaves, that's why it's papaya leaves. Next, protease is used to tenderize the meat. Or you can say soften the meat. Tenderize also can, soften also can. If you put too long hour, your meat will become even too soft, means even more soft. If you put too long, it's going to be very soft lah. Okay, so this is chapter 5. We learn about application of enzyme. Anybody has question for chapter 5? Everybody okay? Okay, then you raise your hand, okay?